Thanks. At first, I'd like to start with thanking the organizers for having me here. And I'd like to stress that my talk is kind of different from what Andrei is given. His great talk, because I will not discuss my own scientific projects, but rather I'd like to give a broad picture of how omics have succeeded in personalized medicine. So let me start with some history. In 2006, I just graduated from a university, and I began my career in biomathematics, and HUPO was my first scientific conference, and its title was moving proteomics from bench to bedside. And I was really excited that as a graduate from medical university, I can help people to treat diseases. So to translate omics to bedside, that's what I wanted to do. And at the same time, here's, by the way, my photo of this conference. At that time, in our institute, we were doing pretty much the same thing. We were researching the ovarian cancer. So now a lot of time had passed through, and now let's see where we are now. So if you take a look at the number of publications in journals regarding omics, you can see here the steady growth. And very important note is here that the scale is exponential. So basically it means that every year the number of articles grows exponentially, and we see the genomics, and recently we see the increase in multi-omics. So from the scientific point of view, everything looks great. There are a lot of articles. Let's see how it's done in practice. So the personalized medicine is tightly connected with omics. And for example, I found here an article which did a systematic analysis of more than 600 articles, and they made a definition of personalized medicine. And personalized medicine seeks to improve stratification and timing of healthcare by utilizing biological information, biomarkers on the level of molecular disease pathways, genetics, proteomics, as well as metabolomics. So we see that omics definitely plays a great part in the personalized medicine. And everybody remembers those bright promises that omics will revolutionize their medicine, and the future will be great just in 5 years. Just let's wait. Omics will change the game completely. So let's talk about each omics, just a few words. I forgot to say a disclaimer that my analysis will be not deep. I'll just go shallow and give you an overall picture. So if you look at the literatures, they say that personalized medicine is approximately equal to genomics. So you can basically omit all other omics and say that genomics is the personalized medicine. And indeed, if you look at the number of genetic tests which we have registered, there are more than 70,000 genetic tests registered, and most of them are diagnostic. Of course, this is because of the great progress made by NGS technologies, and each year there are thousands of new tests being registered. So genomics became a routine, really. So what are the main fields that genomics does? It's pharmacogenomics to individualize drug therapy, and there are a lot of biomarkers for pharmacogenomics listed on the FDA side. For the rare diseases and for newborn inherited disease screening, the genomics is also very good. And their NGS and whole genome analysis can reveal mutations when traditional methods cannot do it. Also forensic genomics. And one of the biggest parts of genomics is precision oncology. It can be said that it's the most successful area of pharmacogenomics, and there are several NGS-based multi-gene panels which are FDA approved. And most importantly, there are clinical trials on the way that use these panels to test whether these new approaches to treatment of patients, whether it indeed gives an increase or in say quality of patient lives. And there are a lot of trials, and some of them are finished, some of them are still in progress. However, I would like to say that the results are somewhat mixed. Some trials failed to show the significant increase in the quality of life, while other clinical trials indeed performed great. So for the transcriptomics, we finished with the genomics, and as I have said, that genomics basically can be treated as a whole or personal medicine. But what for the other omics? So when we look at the transcriptomics, and the most successful story of transcriptomic in every paper, in every book, is MemberPrint, and it's basically very typical success story. So this is a test based on microarray. It uses 70 genes, and it's being used to assess the risks that a breast tumor will metastasize in a few years. And an important note here is that it was initiated very early. Yes, it was started in 2002, so more than 20 years ago. And there are several similar tests available on the market and approved by FDA. And also importantly, there are several clinical trials which indeed show, yes, that MemberPrint works, and it works well. So yes, this is true success story of how transcriptomics can translate to the medicine. Yes, but broadly speaking, this is the main success. Yes, so we have for sure other tests, but they are not so successful as the MemberPrint. So transcriptomics is great, but examples of few. For the proteomics, which I personally have really spent some time trying to do this. Here's an example. The paper by Patrick Cohen was published in Lancet. Yes, the top of the top. Yes, and they used the mass spectrometry-based methods to detect the ovarian cancer, and they were able to achieve perfect sensitivity, and other figures are very good. It was also supported by several other papers. Yes, so the mass spectrometric peak that could discriminate between healthy and normal patients. But later, they were planned to go to the market, but in 2008, they say that the clinical translation had been hampered by sample collection bias, laboratory to laboratory instrument variability. That's all. So until now, we do not see great translation of proteomics into the medicine. As for the epigenomics, we have some tests which are available on the market, which measure the methylation of few genes, and they are also approved by FDA. But this cannot be said as epigenomics because it's the measure of a couple of genes. Yes, there are several promising assays in development, but again, they have to be checked by clinical trials. And however, we see that there's a great increase in the number of clinical trials involving methylation in oncology, and the focus was on colorectal cancer. So it seems it's promising, but let's wait and see. A huge area is epigenetic logs, yes, to predict the biological age from epigenetics. It looks very promising, but still it's not in clinics. So we've talked and briefed about each main omics. Now let's talk about multi-omics. Yes, let's try to combine all this omics data and have our great mathematical methods to combine it and analyze. And let's do some great clinical stuff like disease risk prediction, biomarker discovery, molecular insights, whatever. So this is called system medicine. So when you integrate all the longitudinal data and try to use it in medicine. So the great example here is the pioneering paper by Michael Snyder. So they found that they did a multi-omics profile, and they were able to save him from diabetes, really, because of their proper lifestyle change. But the result of the study is that the most clinical utility was the continuous glucose monitoring. Genomics helped because it found the genetic disease risks, but all other omics were pretty useless. So later we have kind of that's a more recent study that's kind of new Snyder-ROM. So again, a lot of omics, and now it's more than 100 individuals. But what if we look at the results? Are they clinically relevant? So the results basically are mostly scientific. So yes, they found a lot of correlations, which may be of interest for biologists, but not for the clinician. The actionable findings, almost none. They were unable to do so. All these omics data, they were not able to kind of save the patient's lives. Yes, all that was translatable to clinics is some mutation that helped the patient with hereditary hemochromatosis. So the overall picture is translational omics seems to be not very successful. So this is a figure from a paper published more than 10 years ago. That's really we have less than 100 biomarkers. And here's a graph. It shows the number of papers and the number of tests being translated to clinics. But if you see at the number of tests, it's the cumulative number of tests. So in fact, it doesn't show this growth. In fact, it should be pretty constant. So what's wrong, and how we can try to modify things? So I really like this phrase that biomedical scientists are addicted to data like alcoholics are addicted to cheap booze. Sorry. So really, we use the NGS data because it is cheap and we can analyze it and we know how to deal with it. But as Andrei pointed in his previous talk, the environment for sure is really as much as important as genetics and all other things. So maybe we should focus on deep phenotyping and consider lifestyle and environment. So yes, we can measure the molecular phenome, but what about exposome and clinical phenome? They are for sure as important as other things. The most popular application of omics is cancer. But again, what if we move from cancer to other diseases? Maybe we will be more successful there. Another thing is that the variability of omics profiles in healthy people is definitely understudied, and this may be again the cause that the omics biomarkers are not very good translatable to clinics. We have the reproducibility crisis. It's well known that almost 50% of every paper is irreproducible, and a lot is being owed to the bad analysis. And here are some bad advices what to do if you want to cheat. So remember to tell a good story, because everybody likes positive results. And also do some p-hacking, because if you obtain the value of say 0.07, you will try to do everything to make it below this threshold, and to make your boss satisfied, to obtain the grants, and to tell a good story. So there are a lot of things which we need to change, not on the instrumental level, but at the level of data analysis also. And I really like this phrase. So if you torture the data long enough, it will confess to anything. Really, everybody does this. I myself do it. Let's at least think that it's not good. So the artificial intelligence is definitely our new hope, and we can see the great progress being made in the field. And ChatGPT, everybody's aware, and we are thinking that if we put artificial intelligence here in between, maybe it will give us a new push to the bright horizons, right? Yes, that's definitely what we should do. But let's not forget that artificial intelligence sometimes may also fail. And here's an example of IBM Watson project for oncology. So IBM, big company, but it turned out that the project failed. So it produced often inaccurate recommendation that poses serious questions about the process of underlying technology. So do not forget that it also may fail. Recently, there appeared several scoping reviews which try to summarize, systematically summarize studies which were successful in terms that they were later translated to clinics. And out of more than 300 papers for biomedical discovery using machine learning, here's a table that lists only few of them. You can see that not more than a dozen has made its way to a clinic. So what are the features of the successful omics studies? Is that the study design and sample size is really important. If your sample is size is less than 50, your study likely will not make it. The statistical validation and the cross validation is really very important. It is also really important to know what you are going to do, to formulate it clear, to make it available for to make it clear for the doctors. Yes, to specify the inclusion-exclusion criteria and to clearly define the primary and secondary outcome. As I've said earlier, to make the research more reproducible, it's important to have the documentation. Yes, so each time you rerun your pipeline for analysis, it should produce the same results. Model interpretability is an important thing. So in this way, the black box of artificial intelligence doesn't give this human interpretable models. And also they stress the integration of prior knowledge in the form of pathways or ontologies or other things. So I think my time is done, and let's move to some conclusions. So early promises that omics will revolutionize the personalized medicine, they turned out to be overly optimistic, but maybe all promises are overly optimistic. Remember, in the 60s, everybody was sure that we will be on the Mars, but where are we now? And it seems that the most low-hanging fruits are already picked. Another thing is that omics should prove that it is better than traditional methods, and the only way to do it is by clinical trials. Clinical trials is a long and costly process, and it really takes a lot of time and money. One thing that I haven't said in my talk is that there should be more omics in medical education to make physicians acknowledged of what omics can do. But overall, the application of omics in medicine really is a big thing to do, and there's a lot of things to be done. Currently, we're only watering our beautiful tree. It is not yet full grown, so in order to collect the apples, there are still a lot of things to do, but that's very interesting. Let's get to work. So basically, that's all I wanted to say, and so I thank everybody for your attention.
Thank you, Mikhail, for interesting speech, and we have questions. question. First of all, the question from Vladimir Paroykov. How do you define the healthy people? What are the criteria for differentiation them from unhealthy ones?
Oh, that's an interesting question. Unfortunately, myself, I'm not a physician, so I don't know if you ask any physicians. He says everybody. If you search, deeply enough, maybe you can find some traces of disease in everybody. As for myself, I'm just recovering from flu. As maybe I apologize. Maybe it's hearable in my voice. So as for me, definitely, I'm not a healthy person today. So it's really an interesting question to discuss. But now I have no good answer.
We actually said that it's necessary to study more deeply the healthy people. Therefore, it is necessary somehow differentiate the healthy from unhealthy ones.
Yes, that's for sure. Well, maybe if we look more deeper in children, maybe this is the point. Yes, so 20 years old person, can we say that they are healthy? On average, I think yes. So I think that's more data needed to be collected from all the people. But again, this turns out to the question that scientists are addicted to data, like to cheap booze.
Okay, thank you. And we have another question from Yaroslav Faletrov. Dear Mikhail, thank you for your report. Theoretically, how many days can become benefit if proteomics or methylation analysis will be used to detect a cancer before it can be detected by classical clinical methods? Estimate briefly, please, sensitive and precision in days, numbers.
That's an interesting point. So I believe that according to results of clinical trials for the precision oncology, the average increase in survival is approximately about 1 or 2 months. So we do not talk about years. So the increase is significant, statistically significant, but in absolute values, it's about 30 days, 45 days, I think, something like this.
Okay, thank you. And another question from Alexander Dmitriev. What first steps need to be taken to introduce omics into medical practice in Russia?
I think we're no different from any other countries. So I guess we're challenging. We will be faced with the same challenges that in every other country. So the tests, we should clearly define what we are going to do. We should collect the data from a big sample size. Should have ample sample size, and we should exclude any batch effects. So the usual routines. Yes, the analysis should be reproducible. We should not try to, as I have mentioned, to kind of cheat. Yes. So if the cross validation shows that the accuracy is not very good, then we should report it like this.
Okay, thank you. So maybe